Pleasure to introduce Professor Rees, Professor of Respiratory and Critical Care Technology at Aalborg University, Denmark. His interests are in mathematical modeling and physiological process and their application to solve clinical problems. Stephen is a graduate of the City University of London, UK, and he did computing and PhD in health informatics in there. And so we had some visitors from Denmark at City. They were joining a European project. And when they met Stephen, they realized what potential he had. And they grabbed him. They grabbed him the minute he finished his PhD. They grabbed him to the Denmark. He hasn't come back yet. We tried a few times to bring him back, but we couldn't afford it. It is a pleasure to have him. I've got one anecdotal things to say about him. A few years back, I met a dean of a medical school of Aalborg, I think a School of Engineering and Medicine of Aalborg in USA. And I said, I know Stevens. And he said, oh, yes, I know him. He is brilliant. And then he said also that he's got a share of his company. So he bought some share in Stevens' company. I said, oh, I think I might buy it as well. And then I thought, well, every time I bought a share in a company, the company went bankrupt. So I decided not to buy it. On that note, it's a pleasure to have him to give us a talk on commercialization of medical technology, a description of 3 cases of university research from bench to bedside. Thank you, Stephen. It's a pleasure to have you.
Thank you, Abdul, and thanks for inviting me. It's a pleasure to be here, and I have just as many good stories about you as you have about me. I guess we'll save those for another day. Right, so I hope everyone can see my overheads. I'll be sort of looking back and forth to another screen, so apologies for that. But so Abdul asked me to give you a talk, and I've given a talk to the university before a couple of years ago, and at that point we spoke a lot about mechanical ventilation and control of mechanical ventilation. And he said, well, could you give it a different angle this time, so something fresh? And because one of the mantras of our work has been to try and get things at the bedside, to get them applied, I thought I'd give you a view of our research from a perspective of trying to get it used by trying to get it commercialized. And then I have no legal training or business training such, so it'll be a very personal view of that. But I hope nonetheless interesting, and I hope perhaps a little bit inspiring, that if you have ideas, this might give you a pathway through that maze of trying to get ideas out, because it can be a bit of a maze. So I'm going to talk about 3 particular cases, and the first one up here, they're in 3 branches. This is the group that I'm head of, or respiratory and critical care group, RCare, University of Aalborg. And these are the 3 companies that I've been part of founding: Airway Care, Airway Technologies. And I want to talk you through what's happened with these 3 companies, and how the research ideas have become products, where that's gone from there. Not shy away from the engineering and medical aspects, but to give it this tinge of the process of getting from one place to another and getting ideas out there in practice. So let's start with the mid-care story, which is the most complicated and longest of them. So the stories get shorter as we get through the presentation. So the whole idea here is if a patient in intensive care, lots of different machines to monitor cardiovascular status, machines to pumps to deliver medicine, and a mechanical ventilator to support the lungs and deliver gas to the patient who can't breathe themselves. And the question then is, how do you set a mechanical ventilator? What's the best setting? The thing you have to know there, of course, is what there is to set. Here again is a picture of a ventilator. And you can set one of many different things. Set the oxygen, set pressure or the volume delivered to the patient. You can set how many times they breathe. You can set the pressure at the end of their expiration. So when they blow out, what's the residual pressure in the lung? And if they start to breathe themselves, then you can support their muscular breathing by delivering a bit more pressure. So the ventilator will recognize they're trying to breathe and blow a bit more pressure in. So these are the things you can set. And then we get to the question, how is it best to set them? And is this even a problem of how it's best to set them? Well, we know that intensive care is expensive. We know that demands and costs are increasing, and we know, or at least it was projected, mid 2010s, it was projected that the number of intensivists will be reducing. So the need for automation, the need for some help in this process of reducing expertise in a more complex patient population, aging patient population, is clearly there. We also know that poorly ventilating patients increases mortality. Particularly know that if you give them too much volume in a breath, you stress the lungs, that increases mortality. And we know that if you follow guidelines, if you provide an automated process and rigorous guidelines, and they are followed, then practice improves care. So all of these things we know. So it makes sense to try and automate this praxis. And this was our rationale when starting this project. Could we build a system that would help automate the practice of intensive care? Not just intensive care in this case, but mechanical ventilation. Now, our strategy is kind of drawn on this picture. If you have a therapeutic intervention, in this case, oxygen or pressure or something, then if you know the physiological state of the patient, then you can simulate what's likely to happen with them. And then if you simulate what's likely to happen and you look at benefits and detriments of things happening, then you can make a quantification of what's best for the patient. And because we are mathematical modelers, we then wanted to formulate this as mathematics in terms of descriptions of the lung, descriptions of the blood, so that we could tune those models to individual patients and decide the best therapeutic intervention for the patient. So our first step was to say, can we understand the lung with mathematics? And does this help solve clinical problems? If we think of the first one, can you describe the lung with mathematics? Well, the lung is a physical system that has two functions, right? It can function as a pump, getting air in and out, and it can function as a gas exchange mechanism for transporting oxygen and carbon dioxide. Those two mechanisms are very poorly done together, because the pump needs to be robust and rigid and solid, and gas exchange needs to have thin membranes that are delicate and gas can easily transport. So it's not an easily accomplished task. And if you want to represent it in mechanics, in mathematics, then the pump you want to represent with flows and pressures and resistances to flows and pressures. And the gas exchange you want to represent in terms of gas transport across membrane, diffusion, blood flow to take the diffused gas, gas flow to take the diffused gas in the opposite direction. So very different mathematics. So we set about formulating the mathematics of the lung, and we came out with one idea from that. And that was that if you make small changes of oxygen at the mouth, you can learn a lot about the lung. So we found out that if we mathematically modeled the lung, and I won't go into the mathematics, but yeah. And then if we fit that model to data where we varied the inspired oxygen fraction, you can see on the x-axis, 21% oxygen, that's air, and 100% oxygen. We modified the oxygen for different patients, different people. We could characterize different aspects of the lung physiology. So the top line here is how you and I would behave if you turned your oxygen up and down. So one has a very high oxygen level, about 100% in the blood, and you have to get subatmospheric, below normal air, before your oxygen starts to fall. If you just had heart surgery, 3 days after heart surgery, your curve looks like this. And if you're in the intensive care with a very severe disease, the acute respiratory distress syndrome, your curve looks like this. So by turning the oxygen up and down, you can draw different curves and you can learn about the gas exchange properties of the lung. This is what we found out by mathematically describing the lung. And we published several papers on that in different patient groups, showing that you could quantify what happens in the lung. And this became of interest commercially. We thought that there were a number of interesting applications of this. For example, when people have heart failure, they get fluid up in their lungs, and you give them diuretic therapy to get rid of the fluid. Possibly one could then measure how well the diuretic therapy had worked by simply putting a small mask on and turning the oxygen up and down and seeing how the gas exchange in the lungs had improved. And there were a number of these clinical applications. We felt that people may be willing to apply this technology to develop, to have a machine that would vary the oxygen up and down a little and measure the function of the lung. So we wrote a patent. This is the first patent that I've ever written. You can see down here at the bottom, the date was 2000, which means now it no longer counts. A patent has a 20-year life, and then you lose the rights, commercial application. One should remember that a patent only protects you commercially. People always do what they want to do research-wise, and they can always do what they want to do in the clinic. But it protects commercial implementation of a technology. What you should know is the normal process of making a patent, and I think that's quite interesting, is that often you contact a patent attorney and they do a news analysis. So they will have a look for you. What is your idea novel? And then you submit your patent. Usually you have help from the patent attorney because the language of a patent, in particular the claims, there's a series of claims at the end where you specify what you think is novel and therefore protectable. And those claims usually have a very legal language to them, which one needs help. So one usually has a patent attorney to do that. And then you submit your patent, and then the patent remains secret this period of about 18 months, where nobody sees it. And during that 18 months period, at the end of that 18 months period rather, you can decide then whether you want to go national with your patent. What that then means is whether you want to send that patent to the European Patent Council, the Patent Council in the US, to the Chinese, to the Australian, to the Canadian, also separate. And then it will be translated into different languages and be locally assessed by the local patent committees as to whether they will grant their patent. So then it's no longer one patent, then it becomes a patent in each of those countries and lives its own life. So this 18-month point is critical in the sense that if you haven't attracted investment in your idea at that point, then you won't have enough money to go national in the different countries. And then it becomes expensive going national because of the translation fees, because of the separate assessment of the individual patent. So for most researchers, we're at a university, maybe have a contract department, go and talk to them and say, can I make a patent on something that's valuable? And they will help you perhaps with the initial costs of applying. Then the clock starts and you have 18 months. That 18 months then becomes critical to finding a way forward, because if you don't go national at the end of the month, at the end of the 18 months, your patent falls and then it's publicly available and you no longer have any rights to the thing that you're trying to protect, commercial rights. And then, of course, you have this 20-year period where if you don't support the patent on a yearly basis, or a commercial company does, then all of a sudden you lose your rights away. I think that process is of some interest. I think it's particularly of some interest if you've got a patent and you want to go out to the major companies and discuss with them whether they're interested. I've not had any success with that. In my opinion, major companies much prefer to buy small companies when they've become productive. So anyway, we got the patent, and then we formed a small company called Care. It wasn't originally The name changed when we got investment in the company. And Mermaid Care made a series of products. This one on the left here is not Mermaid Care product. This was our own bench-side attempt at university with an ordinary ventilator and a computer attached to it and a gas module. And we would feed the ventilator with a mixture of oxygen and nitrogen to get any inspired oxygen that we want. And we'd wear a mask and we could test the lungs for that. And we did many studies in that way. With Mermaid Care, they built a system here with a breathing device which delivered oxygen, nitrogen, had flow sensors and oxygen sensors. So it could do automatic calculation. It came with quite a large body because there was a tank of oxygen and a tank of nitrogen in here, so it could do automatic mixing of gases. And then afterwards they made a smaller device for the intensive care where you had a patient on a ventilator and you could simply turn the oxygen up and down. I think we realized after some time that the major problem was not necessarily in understanding the lungs and understanding how the lungs were changing over time. Really was, in particular in intensive care, the mechanical ventilation. And I think our interest in trying to get these products pushed out from the company side and also helping the company at the university side became less. So these products sort of faded away a little bit. And the attention focused onto making the device that could support mechanical ventilation. So we're back to this problem, how to set the ventilator. And for us, what you're seeing here is a picture of the ventilator again. And now this is the device that Mermaid Care now produce, which is known as the care system, which is a tablet on top of the ventilator. And you can't see it here, but I'll show you later. There's a gas device that's behind it. So what happens here is that our mathematical models are in that tablet. Now we have the ones that for the lung, and then mathematical models of the blood and how it carries oxygen, and how it carries carbon dioxide, and pH and the acid-base status of the blood. And then we have circulatory models of the blood flow linking acid base and transport of gases with lung models. So those are all in here. And what happens is then the system provides advice. You can see a little bit of it here. Get back to that in a minute. The doctor or the nurse then change the ventilator settings down here according to the advice. And the patient responds from those changes. And those responses, in a good engineering way, help to then retune the models of the patient. You know, if you want to know how hard a wall is, you lean against it, right? So if you want to know if someone responds well to a ventilator change, you make a change, you look at the response, you retune the models to that response. So that's how this system behaves. And it was based on the models and that first patent and a number of other patents. I want to tell you a little bit more about it in detail because I don't want this to be a big black box for you. So what we're doing inside that system, what our research came from and came into this product, was to try and say, what are the compromises in mechanical ventilation? What are we trying to balance? What's the benefit and detriment? So what we did, we said, well, if you're going to turn the oxygen up and down, the benefit is if you turn it up, you get oxygen to the tissues, right? You don't have low oxygenation. You don't have tissues that are dying through lack of oxygenation. But if you turn it up too much, oxygen is toxic. You may not know it, but if you incubate cells with oxygen at higher and higher levels, they will die more quickly. Oxygen is inherently toxic. So that's the first balance. And then if you're going to apply pressure, well, you need to get pressure or volume into the lungs to get rid of carbon dioxide, stop the blood from becoming acidotic. But if you apply too much pressure or volume, you damage the lungs, you rupture them by volume trauma or pressure trauma, barotrauma as it's called. So there's a conflict there that has to be balanced. And if a patient starts to wake up and breathe themselves, you want them to use their muscles, right? Because otherwise their respiratory muscles will disappear. They'll atrophy rather, like any muscle if you don't use it. But you don't want them to use their muscles too much because then they become stressed, can't do all the work that you're asking them to do and then have a relapse. So there's the balance there and how much support you give them. So we put those balances on a hexagon and we said, oh, we want to make these therapeutic interventions of changing things to give us these balances. And in order to do so, we're going to tune all of this process to the person's physiology. We're going to learn about their physiology. How good is their lung functioning? How well is their blood carrying gases? How much oxygen are they using at their tissues? How much carbon dioxide are they producing? And tune the ventilator response to match the patient, and match the patient as the patient changes with time, as their disease progresses and they get better or they get worse. And the models then look like this. So we had models of the lungs, drive to breathing, interstitial fluid buffering, respiratory system mechanical properties like a pump of the functioning. And I'm not going through all the details here today. This isn't the focus. And then we tune those models to the individual patient so we could find the setting which balance the therapy for the individual patient. And we're back, then we put the measurements on top of that, the measurements of flows and pressures and blood, and that helped to tune the individual patient. That's what's happening in that tablet that I showed you previously. And that's what's happening in this tablet, is that all those models are included. They're being tuned according to the measurements. The advice is being created. The doctor can look at the advice and decide, is that appropriate for the patient? Turn the knobs on the ventilator. The system then relearns about the patient, and the whole thing goes in a cycle for the whole duration of the patient's bedside stay at the intensive care where they're mechanically ventilated. Here's a couple of examples of sitting on different ventilators. Actually, this is all from the same company, never mind. Here you see that mounted on an arm here on this old-fashioned ventilator is the gas measurement device. So this is where it's come to the patient to measure the oxygen and CO2. Here you saw the one there and the gas measurement device on the back of it. The gas measurements devices, we learned a lot from the first phase of building those big ones just to understand the lung. If you remember, I showed you those pictures. So all of that understanding has been brought forward into this machine. So this is the one that we currently at the university are doing research with the company to help evaluate. But just let me show you a couple of patients so you get a picture of how it works. So this is a patient. This is the patient who breathes a bit for themselves. So their current oxygen is 41%, and they're breathing for themselves. Every time they try and take a breath, the ventilator gives them a pressure of 14 centimeters of water. So they don't have to do too much work themselves, right? They generate a little under pressure and then the ventilator supports them. So they regulate their own frequency, but the ventilator supports them with extra pressure when they need it. But what the system identified is that this pressure was too much, that we weren't working their muscles enough. So it says, look, they're at risk of atrophy. You should turn it from 14 to 12. So you get a visualization of the patient. And then it says, if you go to 12, you're likely to get closer to the center of this hexagon where they won't be at risk of atrophy and they won't be at risk of stress. It will be a better setting for the patient. So they get this visualization that they can interpret. They get direct advice on how to turn the knobs on the patient, on the ventilator. And then if you want more detail, you can then click and see the simulation. So here you can see that the patient was only breathing 13 times a minute. So they weren't activating their muscles enough. And the system says if you go from a pressure support of 12, they're likely to breathe much more often, 22 times a minute, which will be good for them. It'll activate their muscles, get them closer to coming off the ventilator. So now they can understand the likely response of the patient. If the patient didn't respond by going from 14 to 22, but from 14 to 17, then the models would be retuned and would learn about the patient's response. The advice would be adjusted accordingly. So we get these pictures of the patient, pictures of the compromises that need to be taken, and the model simulations underlying. Behind that, we collaborated with the company on a series of new patents. You can find all of these in literature, but you can see from the titles where it was where they lie: intelligent medical monitoring, mechanical with decision support for lung ventilator setting. All of these patents, you can see their US patent numbers. So they will have had their 18 months, they will have been evaluated in countries, in this case in the US, and then they get a particular patent number for that country, in this case the US patent. And if you read the US patent, you may find it's different from the Chinese patent. Not that I can read the Chinese patent, it's very interesting to see your patent translated into Chinese and the title there in your name, it's all in Chinese. But they may have very different content in the end, because as you discuss with the examiners, they will mature in different ways depending on the local laws of countries. It's quite different what you can protect in certain countries, what you're allowed to protect and what you're not allowed to protect from commercial development. On top of that, of course, it's not possible just to make a device and put it at the bedside. So Mermaid Care spent a lot of time and money in performing regulatory affairs on these devices. In the EU, you have what's called a CE mark, now called actually a medical device regulation, MDR. It's just changed, where you have to show that the device is safe, and you have to document all the procedures in relation to production of the device and the risk analysis associated with the device. And then if you're going to the States, you need approval by the FDA, the Food and Drug Administration. They come in different layers, and I won't at all pretend to be an expert at this, but what I do know is that you can go for a de novo application where you say this is a completely new device, and then the proof of it working and being safe becomes quite high. You have to show them for the first time this is safe and efficacious for the patients. Or you can go for what's called a 510K application, where you say, we know that a device already exists that does this, and we're going to show that this device is equivalent to that, and therefore has to meet the same demands as that previous device. And if it can show it can meet the same demands, then all well and good. And here is the document I downloaded from the net, so I didn't do it controversial secret, which was Care's US Food and Drug Administration approval of Beacon Care System last year. I zoomed in on a bit so you could see it, and what is a 510K. So it says it's substantially equivalent to another device, and that allows Mermaid Care to market the device in the US within the regulatory standards of the US and within the specification of the intended purpose of the device. You need to specify an intended purpose, and then all marketing material, of course, has to follow that intended purpose. So there are very rigid and strict rules around this. It's not university work, I would say. This is work that's done in a company setting. But of course, our collaboration with the company has meant that we've been able to advise on scientific content and description of the work necessary for their application. These collaborations then become very productive for everybody if you can generate such a collaborative environment around these things. Today, we have the Beacon Care system in a number of hospitals in London, Denmark, Italy, France. Excuse me, there's the Danish names of the countries here. We're currently doing randomized control trials, so the system advice is applied on half of the patients and the advice switched off on the other half. We collect data on all of them. In total, we're looking at about 800 patients, and in total, we're looking at all of their stay on mechanical ventilation. So for some patients, I mean, we have COVID patients, they're on the ventilator for 30 days, and we have every breath wave. So it's quite unique data, with the advice being followed for half of the patients. Some of these studies are reaching the end point now. Some of them are still during the progression. I can't talk about the results because they're simply not public. Not even the people working with machines know what the data looks like, because there's research staff are quite blinded to the activities. Because I'm not at the bedside, I'm not a doctor, I'm not blinded to the data, but I have to keep that data secret from people who are at the bedside so that they're biased, influenced by me knowing how the systems behave. the data comes to me, and there we have to follow all of the GDPR regulations on data transfer, and we have to have data sharing agreements and what we're allowed to do and not allowed to do with the data. Of course, it's pseudo-anonymized, so it has to be stored safely. And then we have a collaboration with the company, which generates all of its own needs for managing that cooperation properly, so we don't get into trouble legally. But the university has collaboration agreements with the company, which if any of our ideas can go to the company, and the company provides us with research funding. And the company also do all regulatory work, of course, which allows the device to be used and studies to be performed, which benefits us as researchers, benefits the clinical researchers as well as researchers. And of course, you can imagine this is a hugely expensive process because there are 200 nurses on the site to be trained, turnover is rapid, and the rest of it. So it's not a trivial process to do. And it's been supported by a lot of European money. I converted the money today. I hope I got the conversion right. I calculated 7 million Canadian dollars to date from the EU to support these studies that are running. We've been very involved with the sites of training. Quite exciting. So that's where that stands today. And of course, I can't talk about the company's sales strategy or whatever. I don't know it, and I wouldn't be proper for me to comment either. Just giving a perspective from the university. So that's that story to date. The story to date is that the system is being now largely scale trialed, and we watch the space on whether that'll become an important product, an important device. That's the first story, and I've taken half an hour now. But Abdul, don't worry, that's the longest of them. So I won't be using an hour and a half. The next story.
That's funny, Stephen. But the only thing is, when you turn around, your voice cut off. Just
I'm sorry.
Your microphone maybe need to be adjusted to.
Well, I'll try not to turn around, but if it happens, please tell me. Please tell me. You know, what I'll do then in that case. No, I'm sharing the other screen, so I can't do that. Okay, I'll just try and direct it. I forgot my headphones today. It was unfortunate. I forgot to bring them home. So, yeah, please tell me. So the next story is that of OB Medical. And that came from an idea that you may know that, for example, patients who have chronic lung disease, what happens is often they come into the hospital, are diagnosed, and they go home and they're managed at home. And then they have periods where their disease becomes worse. We call them exacerbation periods. And then they get admitted to hospital. And when they get admitted to hospital, you need to monitor their state. And you monitor their state by measuring arterial blood gas. And what you do to measure arterial blood gas is you puncture their arteries. Now, arteries are thick, they're walled, they're non-elastic, they have high pressure in them, and they're close to nerves. And so they hurt when you puncture them. They hurt quite a lot, actually. And this is quite a cause of anxiety for patients because they need to be punctured frequently. On the other hand, for all of you who've been to the doctor and have blood taken, probably at the elbow, it's a fairly painless process unless you like those sorts of things. And that's quite simple. So because we'd been building these mathematical models, we felt that the mathematical model of blood might be able to help us in mathematically transforming a venous blood to an arterial. And then people wouldn't need to be punctured arterially. And then we could just take venous blood and convert it mathematically to arterial. We were inspired a little bit by the study you can see on the screen by Turner, who looked at intensive care patients and found that it was quite an old study now. They found that the things patients could remember that they didn't like were arterial punctures, and when they had the tube put down their throat to suck mucus from their airways, right? And I'll get back to that because that's the next story I want to tell you. So the two things that they could remember as being the worst complications. So what we did was we developed this mathematical approach. So you can see here that one took a venous blood and one calculated back through the tissues until one got to the arterial. The trick is if you have one of these finger clip pulse oximeters, you get a reasonable approximation of arterial blood oxygenation. And that means if you know the venous blood and the arterial, you sort of know how much oxygen you've removed from the blood as it's gone through the tissues. And then if you say that, you know, the respiratory quotient is 0.82, right? So that the ratio of oxygen taken to carbon dioxide produced is about equivalent, then you know how much carbon dioxide you should add back in, and then you can calculate everything. So that was the mathematical trick that we used to calculate back through the tissues. It's a very simple little calculation if you have an acid-base model of the blood that you can play with, which we did. And that was our model that we used. And then we went through a series of studies to see if it worked in the intensive care patients, to see if it worked in patients in lung medical departments, to see if it worked in emergency medicine with patients with chronic lung disease. And we found out pretty much all the time that the values of measured carbon dioxide and calculated were highly correlated. This is the Outland plot. I'm not sure if you're familiar with these, but what it does is it plots this is the true arterial CO2, and over here is the difference between the measured and the calculated. So what you're looking for is a line that's around 0, right? But these limits here, call them the limits of agreement, and it shows that in these cases, it's highly reproducible. It's good enough to take venous blood and do a mathematical conversion of it. And we're still doing these studies today on different patient groups, but we're convinced now that that this is a sufficient technique and can replace arterial for certain clinical situations. Here's our patent from 2003. So again, we did this, we patented it, and you need the patent because companies, if you want to make a company or you want to sell it to a company, they need protection of the idea, otherwise it's open for everyone and doesn't give them a competitive advantage. This one is nearly 20 years old, so it'll be open at some point, I guess. And again, we funded a small company, OB Medical, and again, I have no control then at all, and nor should I. It has a proper CEO and a proper technical development, and they obtain their own investment from outside and run it as a proper company, and then collaborate back with us as a university partner, where we try and support their activity and they try and support our activity with financial research funding, of course. And that again has worked very nicely. And then last year, that company was purchased by Roche. So now it's called Roche VTAC, VTAC being venous to arterial conversion. And if you go in on the webpage, you can see their videos of the VTAC conversion. And I thought I would show you that because it's a bit fun to see. Start this, show you.
Arterial blood draws and risk of side effects. Roche VTAC works with a range of existing blood gas analyzers on the market. And once implemented, it is simple to operate in daily clinical practice. First, measure the arterial oxygen saturation with a pulse oximeter and collect a peripheral venous blood sample. Then analyze the venous blood sample as usual and enter the arterial oxygen saturation on the screen. The Roche VTAC software then calculates arterial blood gas values, which automatically and instantly are printed on a network printer and sent onwards to other lab systems. Roche VTAC.
There we go. I didn't want to show you it all, but and this is a lot of fun for me, right? It's a lot of fun to have the idea, and then it comes out, and then it's bought, and then people are making commercial videos about it. It's a lot. So anyway, that's been purchased by them, and as far as I know, and we have other research collaborations ongoing with them now because of this. They are marketing this and trying to bring this out into the world. I know that in our local hospital, in their emergency medicine department, one of the local hospitals, they use it on a daily basis so that they use it as a screening procedure because they take lots of peripheral venous blood for other reasons. And then instead of, if they believe the patient might have a respiratory problem, instead of taking arterial, they just do a conversion. And if conversion looks bad, then follow it up with an arterial puncture to check. So they use it as a routine screening tool in emergency medicine. I know a number of other hospitals that are doing the same, but I don't have a perspective on how far this is being pushed out into the market. That's that story. And I have one remaining story, and this is a very new one. This is a company that we formed or so ago called Airway Technologies. And you can see here, I'm back to this overhead from the previous one, but I've marked something red now because we're back to the problem. The second worst problem that was found in 1990, a long time ago, was this tracheal suctioning. I'll explain to you what that is. This is an endotracheal tube. So this is what you get put down your throat, past your glottis and into your trachea if you're mechanically ventilated. And you see on the end of it here is a balloon, and that balloon is connected by an air channel up to an extra little balloon here. And then you put a syringe here and you blow air, and it inflates the balloon down here so that the tube is mounted in place, okay? So that it can't fall out when you're being ventilated. But what happens, of course, is that normally when we're alive, we get secretions building up in our airways, and those secretions get gradually flushed up by natural processes. You have fiber on your airways that wash it up into your mouth, and then you can swallow it. But of course, if you have a blockage of a cuff there, this all sits underneath. And eventually, if it's all sitting underneath, it will block the tube and you can't ventilate the patient in. And then what happens is the nurse comes around with what amounts to a small vacuum cleaner, right? And they put it down your throat, and then you sucks all of this up, right? In quite an unpleasant procedure. So a friend of mine in Italy, a guy called Alberto Zanella, had this idea. It's not my idea. He had this idea that if during a mechanical ventilation inspiration, you deflated this cuff, then instead of the air from the ventilator going down to the patient, it would blow around the cuff. And if you blew around the cuff when it was deflated, you'd take the secretions with you. And then you could inflate the cuff again very quickly during the inspiration. He built this little prototype of this, and he published a study about it, animal models, where he showed that he could effectively remove secretions. And he wrote a patent about it. It's Alberto. There's a number of other inventors, but it's Alberto who I know. And I met him, and he said that he had this patent, didn't know how to move forward. So we discussed, and we felt that we could do something together with other people I knew. And so we founded this little company now in Denmark with his idea, him as a partner, like me as the research partner on this idea. And what's been built, you can see it on the other picture, is this little box here called tracheal flush. And it has a little pump inside that when it recognizes the start of an inspiration, when you push the button there, during the inspiration, it deflates the cuff for 200 milliseconds. And then the air gets blown around, and then the cuff reinflates. And that's the status of it now. We have a number of studies running through ethical approval. We have one study started running through ethical approval to evaluate this technology at the bedside. They CE-marked it by doing good safety analysis with it on different sorts of models to understand whether pressure regulation was correct, make sure it wouldn't damage the trachea if the cuff was over distended, and all sorts of different types of safety tests which allowed them to CE mark, which allowed us then to proceed with clinical studies. And those studies are undergoing. But what we hope is that we can replace all of this suctioning of the tracheal secretions by the simple cuff deflation and inflation from the press of a button. So we're quite excited about how that one will map up. This is a very new store. You can count the number of devices produced here in 10 20s perhaps. And this is their page, travel.com. Talk about secretions from mechanically vetted patients. But again, this company runs now as an independent company, not a member of the company, other than being actually I'm a board member, but I'm not part of the daily running of the company. They work with us at the university. Then we have exchange of ideas and exchange of research financing, of course, in the other direction. So university is happy, companies are happy, relationships are very nice. We have quite an enlightened university in that respect. So that's the 3 stories. That was 45 minutes, Abdul.
That's perfect.
That was
Absolutely perfect.
Oh, I wanted to leave you with some final thoughts. And these are very, very personal final thoughts. I know this is being recorded and released, so I don't want these interpreted as facts. I want them to stimulate a discussion out from experience. So here's the problem. The problem is you have an idea, you want to get that idea, and you need a team to do so, right? It's not just about knowing the technology, it's about knowing the market, it's about having money to do it, it's about being able to regulate it, it's about being able to pay. And if you go to a company with your idea, very often big companies won't want it. And again, this is why I say it's not truth, because very often they will, very often they won't. But my experience is what big companies want is to buy small companies. They want the company who has sold 3 or 4 devices or so and has a potential for growth. They can bring it into their framework. So, and they want ideas that are protected. So then you have to do your IPR, right? You have to get your patents, because if it's not protected, it's open for everyone, and then people don't want it. Well, why go to a company at all? Why not just release the idea? Well, the problem is you can't, because you can't get a medical device to the bedside without it being CE marked or FDA approved. So you need a commercial collaboration. Otherwise, your ideas remain in the lab. Again, I'm sure some of you will disagree with that. That's fine. This is just my experience. So, and to do regulatory approval, which I don't believe is university work, you need to have financing for it, and you need companies for that. So our thoughts have always been, and I remain so, that if we want our ideas to be used, we have to get them out in a form where they can be regulated, where they can be marketed and driven. And it's a bit of in some ways, a negative conclusion, isn't it? Because we'd like ideas to be free and open to everyone, and we'd like devices to be free and open to everyone. But it's an irony that if you make your ideas free, maybe you're less likely to have them used because no one's willing to take them on board. And that's the irony we have to live with, I guess, as technologists and technology developers. That there maybe isn't the altruistic path that we would like, but the other paths are good, and it can be done. Hope you see something. So I'll leave you with that page up, then maybe that promotes some discussion.
Stephen, thank you very much for very excellent and thoughtful and exciting talk. And getting a rush coming and buying a company is brilliant. So it's just must have had a great feeling. So on that note, I'm going to ask our audience if they have questions. Can they